Well, welcome back, everyone. Hopefully you've reviewed the genetics vocabulary I asked you to review, and the purpose of this video is to review the use of punnet boxes. So let's take a typical genetics problem. Let's say our problem is a blue-eyed person mates with a person who is, and we'll just say this is the female for the sake of argument. Let's say a blue-eyed female mates with a male who is heterozygous for brown eyes. Now, in humans, the allele or the gene for blue eyes is recessive to the allele for brown eyes. In other words, the allele for brown eyes is dominant over the allele for blue eyes. So once again, if someone has blue eyes, you know they have to be homozygous dominant. They have to be. If they have brown eyes, they could be homozygous dominant or heterozygous, but in this example, I told you this guy was heterozygous for eye color. So what we're going to do, we're going to say this blue-eyed person, oh, let's keep it legal. This blue-eyed person marries this brown-eyed male. This blue-eyed female marries this brown-eyed male. And we want to know what's the probability that they will produce a blue-eyed child. It is a misconception that dominant means there's the higher probability that that allele will be passed down from generation to generation. The only thing dominant means is that in a case of a tie, it wins. The only thing dominant means that in the heterozygous condition, that is the gene that is expressed. That's the only thing that dominant means. Dominant does not mean it's more common in the general population. Dominant does not mean it usually wins, or it's usually passed on to the next generation. Only thing that dominant means is that when the genes don't match, when there's one dominant and one recessive, the dominant is the one that is expressed. Now, a punnet box is designed, it's a tool of probability, and that's one thing that a lot of, unfortunately, biology instructors forget. It just drives me nuts. But we will go through it. All right, so the first thing we do to do a genetics problem, let's say, so we know what the genotypes of both parents are. As we already established, one parent is homozygous recessive, the other parent is heterozygous. The next thing we do is we decide on the possible gametes. All right, we said she was the female and she can only make one possible ovum. All right, if this is a human female, she will ovulate approximately 500 times in the course of her reproductive lifetime. And I assure you, 100% of the ova will carry the recessive allele. Why? Because that's all she has to give. Now, some people like to make and draw another egg, but that doesn't mean anything. See, this is where I go a little crazy. All the woman can donate to her ova, are the recessive gene, all right, because that's all she has. So 100% of her ova will carry the recessive allele. Now, how many possible sperm can this fellow make? Well, he can produce 2 possible sperm, all right? One or half of his sperm, 50% of his sperm will carry the dominant gene, and 50% of his sperm will carry the recessive allele. Now, I'm not saying this guy is only making 2 sperm. No. There are about 500 million sperm per ejaculate, all right? So this guy will make billions and billions and billions of sperm, not just 2. But 50% of them should carry the dominant gene, 50% of them should carry the recessive gene. All right, it's a 50-50 chance which one he gets to pass on to his kid. So again, big B being dominant doesn't mean most of the sperm get the brown-eyed gene. No. And what color eyes do these sperm have? Well, sperm don't have eyes, right? Again, we have to differentiate between a gene and a trait. All right, 50% of his sperm will carry the gene for brown eyes. 50% of his sperm will carry the gene for blue eyes. None of these sperm will actually have eyes or eye colors. This is an important concept. All right, are we so far so good? So now that we've established 100% of her eggs will contain this genotype, and his sperm will have 2 different genotypes. 50% will carry the dominant allele, 50% will carry the recessive allele. Again, allele just means a different form of the gene. All right, now we make the punnet box. And we just take this information and recopy it in biology. And so I like to put the eggs on top of the box and the sperm on the side of the box, and you'll see why in a minute. So all I did is I recopied, she had 1 possible gamete, so I put that here, 1 possible egg. And now I'll draw a box because I know we're going to have two possible sperm because we've already decided that. And again, I like to put the sperm on the side because their tails can wiggle and be free and swim, right? It's just my little idiosyncrasy. And now we've recorded one possible egg and the two possible sperm from this couple. Now we play what if. What if this sperm fertilizes the one possible egg? What will the future kid look like, or what genotype will they have? Well, this kid would be heterozygous. This child would get the dominant allele from dad and the recessive allele from mom. And their second possible child, all right, will be homozygous recessive. So again, this couple might have 10 kids. This is not saying that this couple will only have 2 children. All right, no, they're saying of the there's 2 possible kids. 50% of their kids will be heterozygous, 50% of their kids will be homozygous recessive. So if I asked what's the probability of any one of their children having blue eyes, the answer, well, we'd have to look at our 2 possible genotypes, and we'd realize, well, this kid should have brown eyes, but this kid should have blue eyes. So 1 out of every 2 possible kids should have blue eyes. So this is the proper use of a Punnet box. All right, and I'll notice I do call it a Punnet box and not a Punnet square because it doesn't always look like squares. And it just drives me crazy when somebody draws an extra egg here, you know, to make it a nice pretty square. This is misusing it. This has no meaning. 100%, I cannot emphasize this enough. Have you noticed? 100% of the ova will carry the recessive allele. So if you add an extra column, you're just duplicating your work and doubling the chance that you'll make a mistake. All right. So if you took Bio 151, when you work Punnet boxes, again, we're assuming that with this heterozygous individual, that 50% of the sperm will carry the dominant allele, 50% will carry the recessive allele. In other words, in this example, there was an equal probability of being fertilized by the sperm with the dominant or the recessive gene. But what we are going to do in this upcoming module, we are going to apply a Punnet box to a whole population. So this is sort of cool. And when we do that, it might not be that 50% of the sperm in the whole population carry the dominant allele. If you look at the whole population, maybe only 30% of the sperm carry the dominant allele, or maybe 25% of the sperm carry the dominant allele. So all we're going to do in this next module is adapt a basic tool of probability that's used in every high school biology text, even though often it's misused. Anyway, I hope you enjoyed this review, and I'll see you in the next module.